This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 802, recorded on September 7th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Kathy Spindler.
Hi, everybody. Here is 82 Fahrenheit, and we know that 82 is 28 in Celsius.
That's true.
And it's sunny, and we have some nice clouds.
We're not that warm. We're 25 C, but nice blue skies. Very nice. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 89 degrees, partly sunny, Texas.
Texas. It's a little cooler than last week. You were in the 90s,
Yeah,
right?
we're on our way out of this nonsense, I think.
And from Madison, New Jersey, Brienne Barker.
Hi, great to be here. Same weather as Vincent. I know no one's surprised, but it is beautiful out there.
Very nice day. Yep. Beautiful day. Our guest today is coming back, I think, for the third time. He is from the La Jolla Institute for Immunology. Shane Crotty, welcome back.
Hi, everybody. Thanks for having me. Yeah, the weather here is San Diego weather.
What is it, 72 and sunny, right?
Yeah.
Do you get tired of that?
No. Somebody asked me on a podcast the other day whether I preferred Florida or Texas weather, and I said San Diego.
Yeah, well, I mean, San Diego has gone through the Austin growth, right? San Diego has grown for years. It's huge, and Austin is just trying to be that way.
Yeah.
All right. Do you want to do your PSA before we get started, Kathy?
Sure. Just to remind everybody that ASV is still sponsoring town halls about vaccine education. Go to asv.org/education. And you should tell your friends and your family and your colleagues about this. If they have questions about the vaccines, we have experts from ASV who are there to talk about them, answer the questions. And a few of them upcoming are in Spanish again. So those are pretty popular as well. So asv.org/education.
All right. So, Sean, I think you were last on last summer, and a lot's happened.
Wait,
so
I thought I just looked it up and it was November.
That's summer here in New York. Really? It's November?
I
think the first one was last summer, which I know a lot of people actually tell me they remember that one. So that was pretty good. And then I think I came on maybe late November for when
November 22nd, 2020.
Yeah, so that would have been our COVID memory study was public at that point and was
okay.
Well, November seems like years ago. So.
It does seem like years.
Yeah.
It seems like last summer. And then since then we had Alessandro on as well after you. So now we're circling back. We get a lot of requests for you guys, so here you are.
By the way, I have, for the listening audience, I have discovered that, well, everybody who's listened for a while understands that you can't understand the content of an episode from the title. Okay.
Right.
And so trying to search for an episode could become problematic. But I've discovered that if you type TWiV and then whatever thing it is you're interested in, you get a pretty good hit.
That's true.
Probably,
Yeah.
Like if you type TWiV Crotty, you'll get all of Shane's past episodes.
Yes, I do the same. I learned a long time ago to do that. Yep. TWiV and whatever, and you get what, because there are no other TWiVs. There are very few others to confound it. If there are, they're on the page 10. All right. So, Shane, I wanted to just go through a bunch of topics that have changed since we last talked. And I mean, one of them, of course, is boosters, but we'll get to that in a bit. But I find there's a lot of discussion about immunity after X, after natural infection, after vaccination, after infection and vaccination, after vaccination and infection. So let's try and get through some of those. Let's start with immunity after natural infection. Give us a sense, antibodies, T cells, breadth, longevity, etc.
Yeah, sure. So what we reported was that after natural infection, looking 8 months out, people generally have T cell memory, B cell memory, and antibodies, and that the kinetics of those look like they would probably be maintained for multiple years. And I'd say there are multiple other groups that had nice studies about individual pieces of that. So we did antibodies, CD4, CD8s, and memory B cells, and a number of labs confirmed individual pieces of that. And then I'd say that Rafi Ahmed and Julie McElrath had a really nice follow-up where they did all those things, and they did different statistical modeling. So from their statistical modeling, they were predicting also that, yes, you should have T cell memory and B cell memory and antibodies for years into the future, and that it was stabilizing around 6 months, sort of, so what you saw between 6 months and 12 months was probably pretty predictive of what you might have for a number of years, which immunologically fits with some other things that we've known. And so that's made sense. And that the T cell response is broad, and that the memory B cells have high quality, which I can come back to, because that connects to the natural immunity plus vaccination. What does that immunity look like? Which I call hybrid immunity. Other people refer to as bulletproof, or some other phrases. But I go with hybrid immunity. A mixture of two things.
Can you talk a little about the heterogeneity? Oh, sorry.
So there's a lot of heterogeneity with natural immunity, and so that's one thing that we certainly pointed out that was quite striking. That between two different people, essentially any two different people, you could have up to 100-fold difference in memory B cells or T cell memory or antibody levels, and that those didn't all correlate. It wasn't that the person with low antibodies necessarily had low T cells or memory B cells. They were largely independent. And so, one of the things that we've said is that that's one really good reason to get vaccinated. That heterogeneity it's not as consistent of immunity as you would expect for a vaccine. And so, if you had a basketball game, right, and one person scored 1 point and the other person scored 100 points, right, you would not consider those similar outcomes. And it's not that immunity is the same thing as a basketball game, but those are big numerical differences. And there have been a wide range of real world studies trying to follow how good is natural immunity, and I'd say most of them have found natural immunity has a significant amount of protection, but generally a little less than what the vaccines give over, let's say, that 6 to 8 month window of time. That's what most of them have reported out. And when I most frequently get asked, well, why should somebody who's previously been infected, right? Why should they get vaccinated? My normal answer is really variants, that it's been less clear how good natural immunity is against variants. Usually people with natural immunity don't have antibodies that recognize variants as well as vaccinated individuals. And really the big Manaus outbreak, the Gamma outbreak, indicated that people with natural immunity probably weren't doing so well, right? Because the implications were there were so many people got infected the first wave, but it seems like they were getting reinfected in the second wave. And so that the protection from natural immunity, at least against that variant, didn't look as good as you would like it to be. Whereas, just to go and step to it, with the hybrid immunity, so people who have been previously infected, whether it was asymptomatic, symptomatic, hospitalized, or whatever, if they get, one dose of an RNA COVID vaccine, their antibody titers are 100 times higher against variants than what they had, the day before they got vaccinated, and that they just have better breadth against every variant anybody's ever seen, right? And they can even neutralize original SARS, right? That's how good their breadth gets with just a single one. And that looks like that's because that connects back to after the infection, they generate such good T cell memory and memory B cells that then when they get one dose of that vaccine, that whole library of interesting memory B cells gets reactivated to make all these interesting antibodies that they basically had as a library. So that's it in a nutshell, I would say, for those two.
So I have two other questions about natural infection. One of them is that I think that sometimes people like to try to make comparisons between the immune responses that we see to different viruses. And so when you look at this response to SARS-CoV-2, can you sort of say that it looks similar to what we see with other infections? Does it look very unique, or is it sort of look like same old, same old?
Yeah, great question. Essentially, as far as we can tell, it looks like a normal immune response to a respiratory virus infection that was, going to give you flu-like symptoms, and actually pretty not too unlike what some live viral vaccines have even given you. Surprisingly, when we tried to do the head-to-head comparisons, not head-to-head, when we tried to compare to the literature, we actually found that there's really not many reference points for. We said, all right. We measured T cell memory and memory B cells for 8 months, right, in people after SARS-CoV-2 infection. How does that compare to other infections? And honestly, nobody had done it before. So that was the largest study ever of immune memory over that kind of time window for any viral infection that actually had, longitudinal data. So the bits and pieces we could find was really yellow fever virus data from the vaccine, honestly, from Rafi Ahmed, and the memory T cells look basically as good after a SARS-2 infection as they do against a yellow fever vaccination. So that's a pretty strong, reference point. And then for memory B cells, there were about, I think, half a dozen people that Laura Walker studied with yellow fever vaccine actually again, and actually looked kind of similar, in terms of number and quantity. So overall, it looked good, though we didn't have as many benchmarks as we would have liked.
Yeah. And my other question is about compartmentalization. So, most of what you are showing here is blood. Do you have any kind of thoughts about whether you see kind of that same great mucosal protection, same great memory protection in the mucosal surfaces, or things like that?
Yeah, that's absolutely the biggest gap in knowledge still is, it's absolutely appropriate for somebody when talking about immunity to say, look, it's like real estate, right? Location, location, location. Where is it? And for vaccines, we can basically say, look, location matters, but for lots of things, when you generate that circulating immunity, right, that immunity in the blood, it can get to where it needs to get in a reasonable amount of time. And that looks to be, that's been the case, I think, for SARS-CoV-2. And that what you talk about the most with the vaccines, right, is can they prevent the hospitalizations and deaths? And all of that's about lung infection. And, hospitalization numbers we talk about are hospitalizations like day 21. That's a really long time after you've been infected. And so there are weeks for the cells to get to the right place and do the right thing. And I'm sure they get to the right place and they're doing something useful at least by day 6, and so the fact that it's circulating and not at the portal of entry is okay for those metrics. But for natural immunity, there's going to be more of a bias. We would expect more of a bias in location, but there hasn't been. There's still a knowledge gap there. There's some data on antibodies, that they're measurable, and particularly in saliva, which is easier to get samples from than the nose. Saliva data out for like 6 months post-infection, but it's still a real gap. I wish we had better data.
So what about after vaccination only? How does that compare with natural infection in terms of longevity and breadth? And you've looked at mRNA vaccines, right?
Yeah. Yeah, so the first available data was for Moderna vaccine, really done by the NIH group and Mehul Sudar at Emory, seeing that 6 months out from in the clinical trials, they definitely still had antibodies, and it was about a 5-fold to 10-fold decline, depending on which number you use, which is a significant decline. And so then the question really kind of becomes, is it stabilizing around 6 months, or is it going to be pretty similar 12 months, or are you going to continue that type of decline? And so we were the first to report T-cell memory for an RNA vaccine, which was medRxiv maybe 6 weeks ago. And we actually did it for the 25 microgram Moderna vaccine, the low dose Moderna vaccine. And we thought, well, with the low dose vaccine, that's actually kind of a higher bar. Did you actually generate good memory after, even if you got a lower dose of the vaccine? And we were actually really impressed that there was the T-cell memory was quite stable out to 6 months after the second dose. So 7 months overall. So 6 months out from the second dose. And there was only a 2-fold decline from peak for the T-cells. It looks, it looks quite stable. It looks like it would last for years, which again, we were really impressed by because there really hadn't ever been RNA vaccine data like that. We were worried that maybe it didn't generate good memory, and it looks like it generates great memory, both for the CD4s and the CD8s. And John Weary's now got a really nice preprint out where they've looked predominantly in Pfizer vaccine, and they see good T-cell memory and good memory B cells. And so overall, to me, it looks like the RNA vaccines are generating quite good immune memory, good quality, good magnitude of memory B cells, CD4s and CD8s. And so I would expect that would mean, and Ali Alibedi published that he could see long-lived plasma cells after vaccination. So there's at least some long-term antibody production that's going to go on. So I would expect antibodies to kind of stabilize, between 6 and 12 months, given the quality of the other memory. I guess I'll pause there.
So I'm asking this question mostly because a lot of people ask this question of me. And I want to, kind of see what you think for the answer as well. So with that response to vaccine, how much variation do you think that you might see when varying the interval between the first and the second dose? Would that make a big difference in the big picture? Would it make a difference in the details? What do you speculate you might see there?
Yeah, so so it's actually a topic that we research actively in vaccine immunology in my lab, and it's actually something in the HIV vaccine field that I pushed on like 5 years ago. That essentially all the monkey immunization studies, it was basically vaccinate repeatedly as fast as you could and vaccinate as many times as you could, and I actually pushed to say, look, what's lacking here is quality in the immune response, and quality is a different process, and it can take some time. And so let's try and give the immune system some more time between immunizations for developing quality. And really, that would be from a T-cell side, giving them a chance to rest, which is kind of classic work from Rafi Ahmed's lab and others, that the T-cells enjoy a bit of rest before seeing antigen again to give best quality memory. And on the antibody side, it's really about the germinal centers, that the germinal centers can go on for quite a while after the immunization is over. And it's in those germinal centers that you're getting this real-time evolution, right? And an increase of antibody quality and competition between the different B cells and antibodies. And it's unclear what happens when you re-immunize. You might actually be shutting off all those germinal center reactions. So whatever was going on, you've now basically truncated it and told it to start over, sort of thing. And so he said, look, just let's try and be patient. Basically, it's really hard to be patient, but let's give it a chance. And so when we space things out to 8 weeks instead of 4 weeks, we actually started getting better, much better neutralizing antibody responses in the monkeys than at 4 weeks. And we've been exploring that even further now in work that we've got preliminary data on.
And that was the HIV studies? Sorry.
That was HIV studies.
Okay.
And so, we said, look, for COVID-19, when people were saying, hey, I got my first dose of Moderna and I missed my appointment, right? And so people, I missed my 4-week appointment, am I done? Can I not? Am I outside of my window of opportunity? Am I stuck with the one dose? And the other, no, that's not it at all. It's that 4 weeks is the fastest you could get that second dose. It's like, get it at 6 weeks, get it at 8 weeks. And I said, even, back in December, I was like, your immune response would be at least as good if you've gone 6 or 8 weeks as 4 weeks, and probably better than at 4 weeks. The Moderna and Pfizer were making decisions based on what's the fastest, it's a pandemic, what's the fastest we can possibly immunize people and get them protection, right? Not, we've got a year, right? What's the optimal, most patient way to do it? And so, I think it'll be really valuable to see different timings done and see what the data are. The clearest data so far has been from one English study that compared Pfizer 3-week to Pfizer, I can't remember if it was 8 or 12 week. And what they saw was the delayed boost gave twofold better neutralizing antibody titers, but actually gave a little worse T cells gave about twofold less T cells by the assays that they used. And so, I think it needs some additional study. I definitely think the timing intervals matter, which is one reason people have talked about boosters, to say, look, what you got done early was things you could get done fast, but giving your body 6 months and then coming back, there's some immunological value in that.
Now, if you don't, without any boosting, how long would germinal center maturation continue? Do we know?
That, Vincent, is a fantastic question, and we've been working hard to answer it. So in mice, it's generally like a 14 to 21 day process. That's an average germinal center. And then if you get really good ones, it could go like 28 days. But in monkeys, when we started doing these HIV studies, we said, look, we just don't know, because germinal centers only occur in lymph nodes. They don't occur in blood. And so, 99% of the time you measure what comes out in blood and you're just guessing. And so we started using an actual clinical technique, lymph node fine needle aspirates, where we would immunize the monkeys, like in the shoulder, say, and then, the lymph node that becomes reactive is the armpit lymph node, the axillary lymph node. So you can actually insert a small needle into that lymph node and just siphon off a small number of cells and just see, are there germinal centers there? And we started doing that, and it was really valuable. And you can go back and you can do it longitudinally. So when we did that, we actually saw they would be having germinal centers for 8 weeks, 8 weeks or more. And it was in those animals that they got really good neutralizing antibody responses. So those are things that we know. And then Ali Betty at WashU has used that same technique now in humans with people getting the Pfizer vaccine, and he saw evidence of germinal centers at least out at 10 weeks in some of those people. Handful of people, but it's.
So you said earlier that infection followed by vaccination, you get basically a better response. How about, to me, that sounds mostly like, okay, it's a boost. Mm-hmm. Okay. So what happens if you get infected twice? Do you know?
Yeah, I haven't seen any study on that. It's a great question. For me, you'd have to break that out into the type of infection, right? Because a number of those are people who got basically an asymptomatic infection first, and then they got a symptomatic infection second. And so maybe they just didn't generate much the first time around. But yeah, it'd be good to see. I think I can't even remember if I've seen a case study on that. I've seen antibody case studies that they certainly get an antibody boost, N = 1 type stuff. I don't think I've seen any T cell data.
So I want to clarify or try to clarify something else up on top, and that is the role of the different types of immunity, antibody, humoral versus cellular. You have a statement in one of your papers. This is the, never mind. It's in one of your papers. While it is not anticipated that circulating memory T cells would be effective in preventing SARS-CoV-2 infection, it is plausible that they can contribute in reducing COVID-19 severity. So you're making a distinction between the arms of the immune system and its role in infection and severity. So I'd be interested in you elaborating on what you're thinking, how you distinguish between infection and severity, and how you think the different arms of the immune response play into those processes.
Yeah, sure. Boy, Vincent, do you ever show slides in this? I actually have.
Yeah, you can share your screen, sure.
I wasn't planning on it, but I literally have it on my screen. Yeah. And I was saying this to the WHO on Friday. So, all right, let's do it. There we go. Just go. Yeah, I mean, I basically, let's see, I'll do it. So, well, you'll like this. I always start with the virology. I was like, look, the parts of your immune system that can contribute to protection depend on the nature of the disease, right? How fast is the disease and where is it? And in this case, we've got a virus that's really fast at replicating, right, in nasal spaces and oral spaces. So within 4 days, you're at a point where you can transmit to other people and you're symptomatic at day 6, right? And it's a race between the virus and your immune system. But the serious disease is all about the lungs. That's where you get the pneumonia, that's where you get the hospitalization, and that's where you get the death. And for hospitalizations, you know, we always talk about hospitalization numbers at day 21 because it's actually a slow infection in terms of getting that lung infection and getting to severity that would result in hospitalization. So you basically got fast infection in these two tissues and slow infection here. And then you've got this spectrum of symptoms, right? And so the nasal and oral, that's your asymptomatic, polysymptomatic, cold-like, and even flu-like. And really, it's the lung infection that are your hospitalizations, ICU, and fatal cases. Okay, and so with that in mind, what's the immunology? Well, the immunology is antibodies are your first line of defense. Period. Right? So if you can have so much antibody present that you can stop the virus, right? Sterilizing immunity. That's all you need. That first line of defense is all you need. And so if you're trying to prevent all infections, antibodies are going to be more dominant. And that's this slide. So when we talk about protection and immunity, we're talking about protection against any detectable infection. That's probably antibodies are playing the major role and T cells are playing a minor role for the vaccines. And I think the correlative immunity study of the Moderna is consistent with that. But if you're talking about protection against hospitalizations and death, or even, you know, a lot of symptomatic disease, which is occurring at day 7 or later, it's much more likely that it's a mixture and that the disease is slow enough that's 7 days is enough time for CD4s or CD8s or memory B cells to contribute. So if you are in a situation where your antibodies have waned, or if it's a variant that your antibodies don't bind to very well, or you never made much of an antibody response at all, for example, because you were immunocompromised in some particular way, I think there's plenty of data supporting that these other components of the immune system can still do a lot that protect you against these more serious outcomes. There you go, Rich. That's the 2-slide summary of my perspective on it.
And vaccination is going to help out in both respects. Okay, and that You've got an antibody response that's going to help on the front lines with the initial infection. But you've also got a T cell response so you got a head start on the lung pathology, so that that T cell response, in addition to the others, can help impact on the severity. Okay.
Yeah. So like the New York State data and the big Kaiser preprint that came out that said, look, out at something like 4 to 6 months post-vaccination, we're seeing waning immunity in terms of cases, right? Maybe going from 90% to something like 50%, but still really great protection against hospitalization, right? Like no change in hospitalizations. People go, well, wait, why aren't those connected? Why aren't you seeing the same numbers? And it's because they're really temporally two different kinds of outcomes. You can still get infected, for a few days, but still have plenty of immunity that's going to prevent the lung infection, pneumonia, and outcomes because of the different parts of the immune system that contribute. Yeah.
So when I tell that to people, some of them will then say, why is it in Israel you got so many hospitalizations, which seems to be different from the experience elsewhere? As you say, the vaccines are still in the 90 percentiles, but in Israel it seemed not to be the case, and I can't explain it.
Yeah, I can't particularly explain it either. I mean, the vaccines, even in the Israeli experience, the vaccines have still held up very well with hospitalizations, but they have seen a measurable decline in protection against hospitalizations. They still don't have a full-blown preprint or paper on that. Where we could dig into whether there are confounders in the math, but no, I can't explain that. But in the preprints that are available, like the Kaiser one, right, where they did take a look, certainly the vaccines are holding up really well for hospitalizations and deaths, even in studies where there's some measurable decline in protection against detectable infections.
I want to wait for the peer-reviewed data on that one, because we had a communication from a listener at one point who's a statistics guy, runs a website, who looked at I believe it's the same Israeli data, and he said, it depends on how you break it out. Okay, and if you tinker with the demographics and that kind of stuff, you get a different set of numbers. Okay, so I think that's going to be important.
And so, Rich, just to expand on my answer, the other way I've answered it has actually been, look, I'm not an epidemiologist, and epidemiology is a science for a reason. You don't just take A plus B and you get C. And what I do know is that there were a variety of different numbers and things people were saying about how good the original vaccines were, in that population. And then it took several months for epidemiologists to go through and actually figure out the confounding variables between age and geography and some other behaviors there, which they talk through in this New England Journal of Medicine paper, right, on the real world efficacy of the Pfizer vaccine in Israel. And it was actually pretty complicated for them to match people up in that particular population. And so, I've leaned more on, there's a nice preprint out of England. There's two nice preprints out of England where they've done a lot of controlling for confounders. And then again, the Kaiser study looks pretty robust as well, where there is evidence of some waning in protection against detectable infections, but not the hospitalizations and deaths, which, I think that's where we are.
Okay. So let's round out the discussion. What if you get vaccinated and then you get some kind of an infection? Everyone wants to know, does my immunity look like infection vaccinated, or is it different? Do we even know?
We don't. And I get that question a lot right now. We're certainly interested in studying. We're certainly studying it. Other people are studying it. It'll take a little bit of time. I think I don't have a clear prediction for it, because in that scenario, you're vaccinated, so you already have a substantial amount of immunity. And so when you get infected, I'm I'm sure there'll be some boost, but I don't know how much of a boost because the vaccine's gonna blunt it, and so it's a little harder to predict that outcome. I do expect that you will end up with some more localized immunity in your nose and mouth as a result, because when you got vaccinated, you got vaccinated systemically, and so your immune system doesn't particularly know where to go. And then if you then get infected, now your immune system, just going to say, oh, that's where the virus shows up. All right, well, let's hang out more there. We'll leave at least a little bit of memory in the nose and mouth. And so I do think that'll be interesting to follow up on, but I haven't seen any publications on it yet.
So a difference between infection, a significant difference between infection and vaccination is that when you get infected, you're seeing the full spectrum of virus gene products. Okay. And as you've quite elegantly shown, that includes a huge variety in T cell epitopes. And there's also a larger variety than in vaccination of B cell epitopes as well. And when you vaccinate, you're getting only spike. Okay. So, I have a hard time thinking about the effect of a vaccination over an infection in this regard. Okay.
Yeah.
If vaccinating an infected person has such a profound effect on their immunity, in particular with respect to variants, when you vaccinate somebody who's already infected, all of those other non-S epitopes, are they not being affected?
Yep, they just sit there.
They just sit there.
It's a nice controlled experiment. You see, those are stable, and then the spike ones all jump, and you're like, all right.
Okay. Okay. So the effect of the vaccination really is just boosting the spike response. Okay.
And so for this virus, it works out pretty well. First of all, spike is essentially the target of all the neutralizing antibodies, and so having only that works out well. And then on the T cell side, while there are 25 different proteins, gene products in SARS-CoV-2, and almost all of them are targets of T cells in different people, spike is a very large protein. And it's quite well expressed normally by the virus. And so what we observed initially was that spike's actually one of the immunodominant targets of the T cells. Most of the T cell responses end up being against spike in M overall, just numerically the total number of T cells, right? And many of the epitopes. And so spike is big enough so that even if you are just immunizing with that one protein, there are actually a lot of different T cell epitopes in it. So you still get a decent, you still get a solid breadth, whereas it's actually a somewhat different equation if you've got a virus with a rather small receptor protein, and that you really may be lacking epitopes, and you may have to go look elsewhere, for decent T cell epitopes. That's not a problem when you have this giant spike.
Is that
so? Thinking again about the difference in the spectrum of the difference between infection and vaccination in the spectrum of epitopes, we're saying here is that infection and vaccination is better than vaccination and vaccination so far, right? It seems like if you get infected and then get a vaccine as a boost, that's better than getting a vaccine and then another same vaccine again.
Well, Rich, right? Do you mean better? When you say better, do you mean something like quality, or do you mean something like quantity or breadth?
Yeah, I don't know.
All of the above.
All of the above. What I'm going
immunological measurements. Yeah, it's been yes and yes and yes, actually, because actually in this there have been two studies. There's a Kentucky study, and then this nice UK Powell study did find that people who had this hybrid immunity, if they'd previously been infected and then they got vaccinated, those people had the best protection. And then the immunological measurements have been that, those people have the most T cells, the most antibodies, the broadest against variants.
So I'm what I'm thinking then is that the kind of extra epitopes provided by the infection to start with, not that I'm recommending this, okay, contribute to this better response? If you've got more epitopes, or is there something else about infection altogether that we're not even talking about that affects the response?
So far, it could all be explained by really that breadth and quality of the memory B cells. That just that you end up with literally 100 times higher neutralizing antibodies against variants with that particular combo, and that seems to happen because after the infection, you generate a high quality T cell response that then helps the B cell response. And then that product of that ends up being these memory B cells that can recognize a bunch of different variants. And then as soon as you get that vaccine, it recalls those memory B cells to actually make all those really cool antibodies that you actually had sitting as sort of a library. I end up talking about memory B cells as, they've got two kinds of value. So one is you've got a certain amount of circulating antibody, right? And so if you get infected and the virus gets past that antibody, well, what's your backup plan? So the memory B cells are the cells that can make more of those same antibodies, right? And so they're sitting there they're like antibody factories with the lights turned off, right? They got the power off and then they just sit there and wait. And if you get an infection, right, that factory turns on, you crank out more of the same antibody. But they have this whole other value. Half the value is that making more of the same. But the other half is there really are this library of immunological variants that the memory B cells are. They all have different individual mutations in them that are guesses your immune system's making about what viral variants might look like. Because a lot of the value of memory B cells is, you made all this antibody, and if the virus gets past that antibody, well, maybe that's because the antibody wasn't as good as you thought it was going to be, right? And maybe it wasn't as good because the virus has changed, right? And your immune system's had millions of years to figure out that, hey, viral variants are a real thing, right? And so making these guesses and having this library, this collection of memory B cells makes it so when you do have a new exposure, you have all these guesses already in place of what might actually work really well. And so with the vaccine, all of a sudden those get recalled and you end up with these really potent, high quality antibody responses. So it looks like most of it can be explained by that. But these other T cell epitopes against other sites could absolutely contribute.
So I have a question about quality of responses, particularly the memory B cell responses. If you compare the vaccination response or the infection response or the combo, in whichever direction you want to take that combo, one thing that I start to wonder about are the data that I saw from that paper from Shiv Pillai's lab about germinal centers and germinal center changes in severe COVID infection, and whether those kind of changes after infection might impact quality. Does vaccination actually prevent you from having that severe infection so you don't have that germinal center decline? Do you actually get a difference in quality with vaccination and infection, as would be implied by that? So how do you think that actually plays out with your memory B cells?
So for one, yes, vaccination is going to prevent those severe outcomes. And so, just intrinsically put you in a much better place. Overall, really, a lot of the best memory B cell data, there's several labs that have contributed, but a lot of the data from Michelle Nusensweig's lab was really good, where they tracked both hospitalized and non-hospitalized, right? And they sequenced the memory B cells and they made those antibodies and asked what's the quality of those antibodies, right? And when you waited out to 6 months instead of 1 month, how did it look? And just overall, the quality of those memory B cells was really good from all of the infected people. So that was certainly a general question people had was, well, after an infection with this virus, do you generate quality immune memory? And that was direct data for the memory B cells that, yes, the antibodies these memory B cells make can have really good quality after infection. The immune response is making quite good germinal center responses to that. And then following up on that became essentially the hybrid immunity studies of saying, well, when those people got vaccinated, they made these fantastic antibody responses to variants. Oh, yep, that's because we've got these studies from Nussensweig and others that show these memory B cells. This is the evidence that those memory B cells really did get recalled when you saw a new exposure, i.e. the vaccine, and made some really fantastic antibodies.
I'm kind of thinking about your analogy of the memory B cells being a factory that the lights come on, but maybe also in the meantime, other assembly lines have been put in place that can then give you that variety, additional optimized antibody. Does that kind of work for you? See what I'm saying?
Yeah, I mean, the idea is each factory is making a slightly different version, I don't know, a version of what? I hadn't been thinking through it that way. I guess different versions of missiles, and so
Yeah.
Sometimes a heat-seeking missile is the way you want to go, and sometimes some other missile is the other. You could do the analogy a variety of ways. Tires that work on one terrain versus another.
Right. Okay.
So let's talk a little bit about boosters. Looks like the U.S. is barreling towards boosters, and I know what you've said about it, and people have different views. So tell us your thoughts. What are you thinking and why?
Yeah, I think so. I think the simplest position to be saying no boosters really comes from looking at the available data and saying, look, we don't see a problem with hospitalizations or deaths. The vaccines are doing what they're really supposed to do. We're okay. And then I think arguments for boosters come from several different lines of thought. And one is, well, if efficacy against cases per se drops from 90% to 50% at 6 months, maybe that portends a drop against hospitalization efficacy in the future by a few months, right? So a better safe than sorry approach may be, get a booster now and reset that clock so that you don't run into that trouble. And so a better safe than sorry approach is reasonable to consider in uncertainties. Another is to say, well, if you're just telling me that there's a flu vaccine, right, and I was 95% protected and now I'm 50% protected, yeah, I would like to get back up to that 95%. I don't want flu-like disease. So those are a couple of them. And then when people talk about cases, and we just say, well, look, cases, it's not that big a deal, right? Like somebody in my lab got sick and they were sick for a week with an upper respiratory tract infection. It wasn't COVID. So we're like, no big deal. But, and my son got sick for like 2 weeks, right? It was something, it wasn't COVID, but it, knocked him out. So we're like, yeah, we got to just get back to getting used to this real world scenario, right? If we get sick with things. But with COVID, currently, it's more complicated than that, particularly for hospitals and healthcare workers. I talk with my colleagues at the hospital and they're like, look, if you test positive for a case, you've got to be out of work for a week, right? And so the hospitals are having trouble staffing. So if their protective efficacies drop to 50%, say, and they've got people showing up positive, now they run into staffing problems. Plus, staff not wanting to come in, right? And that scenario seems to be a problem as well. So those are a couple of the factors playing into it. People have some concern about long COVID, right? Could you still get long COVID? To me, overall, the data look like vaccination should largely prevent long COVID. I think mostly long COVID looks to be a product of severe disease, either because of just the total amount of viral load or disease getting so severe you trigger unusual immunological outcomes. But, it's an uncertainty. So people keeping that in mind in that realm is reasonable. And so, And so then lastly, there are a lot of vaccines that are 3-dose vaccines, and they are 3-dose vaccines because immune memory just isn't that good after 2 shots. That after 2 shots of tetanus or 2 shots of hepatitis B vaccine, you make you make antibodies and then they just fade after a few months. But then you get that third shot out at 6 months, and now you've got antibodies for 10 plus years, right? 30 years for the tetanus vaccine. And some of that comes down to your immune system's doing a cost-benefit analysis, simple as that. It sees it takes a lot of calories. It takes a lot of resources to make antibodies for a decade. That's a lot of energy. And so if you see something once and it wasn't life-threatening, it's worth some resources, but not a lot. You see something twice, it's worth some more. And if you get infected with something a third time, right, it really usually forces your immune system to go, okay, that plan was not working, right? We need to commit some more frontline troops to this, And so there's a cost-benefit analysis that the immune system is doing. And so 3 doses of a vaccine tends to generate longer-lasting memory than just 2 doses. And so the prediction would be you would sustain antibody titers for a longer period of time after that third dose. The reasons not to do it, not to get a booster, would be one, or not to approve boosters, I would say. One would be reactogenicity concerns, right? Just basic safety issues. So the booster clinical trials look fantastic in terms of their immune response. The antibodies are broader against variants. They're boosted back up to probably 2 to 4 times the level of what the second vaccination gave. But it was only about 100 people, right? So that's not a lot of reactogenicity data. So it was safe in those 100 people. My understanding is there's now, they did studies in Israel of a couple thousand people, and the reactogenicity was basically the same as it was to the second dose. So that would match expectations, and certainly from a safety perspective, that would then would be fine. And so then the other concern is really the global equity concern that look, how can you give a third dose to people when there are all these people who haven't gotten a first dose? And I think there are a number of reasonable perspectives on that. I'll say I certainly care about global equity, right? I mean, pre-COVID, I spent my entire decade working every day on an HIV vaccine. But practically speaking, in terms of COVID vaccines in the United States, the U.S. has to retain enough vaccine doses to vaccinate all the unvaccinated people in the country, which is probably close to 100 million people still. You have to still try to vaccinate those people, right? And so you have to keep enough doses around to try and vaccinate those people. And hopefully you convince them, but that hasn't really been happening, right? So then I think if you have to keep those doses around, why not give them to people who actually want them instead of having them expire? I mean, right now we've got millions of doses expiring on a regular basis because they're being held around. I see no downside to allowing people get those as a booster if people aren't getting them as an unvaccinated person. To be clear, a dose of vaccine into an unvaccinated person is absolutely more valuable than a dose into a vaccinated person by any metric. I mean, deaths, hospitalization, transmission, you name it. But if you can't give it to that person, using it as a booster, I don't see it as a loss. I really liked what Carlos Del Rio said about it on the UCSF Grand Rounds we did. He's a public health expert at Emory. He said, look, it's like when I was a kid and my parents told me to finish my dinner because there were starving kids in India. And I said, it's important to care about the starving kids in India, but the dinner on my plate doesn't help those kids at all. It's a false dichotomy. You can't, getting vaccine to other parts of the world is very, very important. But it's a false dichotomy. It's not an either or. It's really an and. That's my take on it.
So one of the things people are thinking about is that, and you touched on this briefly, let's just make it clear. Infection, vaccination, incredible breadth of antibody response. Are you saying after a third shot, you get a similar antibody breadth?
Yes. So both. So in the Moderna trial the Moderna booster trial was first, and in that one they compared their original vaccine as a booster, and then they also compared a beta as a booster. And in both cases, they generated good neutralizing antibody breadth against variants. And then boosting with the original was just about as good as boosting with the beta, even for getting neutralizing antibodies against beta. So yeah, similar type of thing as hybrid immunity, that this is a virus that each time your immune system sees it, even if it's seeing the same virus and not actually seeing a variant, it is learning to recognize variants better.
So you don't really need to boost with variant vaccines, right?
Right. So that's the other thing that certainly people who are like, wait, if we're going to do a boost, why isn't it a Delta boost? And that's a fair question. And one reason is that you have to wait, right? And the other reason is really the Moderna trial was directly asking that question. And I was definitely a proponent of that trial occurring and comparing those things. So like, yeah, now's the time to learn, do you need a variant booster, or is original strength or vanilla or whatever you want to call it, going to get the job done? And yes, it's going to get the job done. And the Pfizer trial corroborated that evidence.
So based on everything that has happened with thinking about immune responses to SARS-CoV-2 in vaccination and infection and all of these things, what do you think we as immunologists have actually learned about the immune system?
Actually, huge amounts, honestly, because a lot of human immunology is restricted by the fact that in any single place in time, you've only got a few cases of a certain infection or a certain disease with a certain complication or even certain vaccination, And so you have all these studies that were 5, 10, 20 people that you were, or even 2 people that you were trying to extrapolate from. And now between the vaccination programs and the infections, you have these studies that are very well powered essentially to answer good questions. So honestly, it's hard for me to think of anything else that we've learned as much about human immunology as we have had with COVID. In part because there have been really good, well-funded NIH-supported groups that had all these pipelines in place, to measure the right things in the right ways, and so that the tools were there. And the other is, what I was just describing, actually having samples available for comparison within reasonable windows of time has allowed for really quite statistically robust conclusions about how the human immune system really functions. People still ask me all the time to predict immune memory, like, what's memory going to be like, 4 years from now? They're like, we don't know. You know why? Because it hasn't happened yet.
So we talk, a lot of what we talk about, your work and others, is mainly with mRNA vaccines, but some of the generalizations we've made today, can they be applied to, say, adeno-vectored spikes or even inactivated vaccines that are made elsewhere?
Or subunit vaccines.
Subunit, yeah.
Yeah, they can definitely be applied to most of those. Okay. Yeah. I've certainly been impressed by the clinical trials that I've seen studying adenoviral, basically all AstraZeneca, and then an RNA vaccine as the second immunization. So first immunization, AstraZeneca, second immunization, RNA vaccine. And a couple of those have actually have had the full comparison. You either get 2 shots of adenovirus or 2 shots of RNA or 1 shot of each. And the mix and match did in fact look significantly better. So there's some of those things that we're learning about as well. It's just there have been so many more people vaccinated in the U.S. with the RNA vaccines than J&J. There's just a lot more limited data. Here at LGI, we're definitely studying the J&J vaccine, and we've just been trying hard to collect enough people at the right time point so that we could make a meaningful comparison.
Great. Shane Crotty, as always, very illuminating. Really appreciate your joining us. Thank you so much.
Yeah, I really enjoy this. I always feel
educated,
immune when we're done. The immune system is really cool. It's it's thought of everything, seems like.
Yeah. Yeah, I mean, the immune system is really good at recognizing and dealing with this virus, and definitely all of these approved vaccines are really good at dealing with it. Definitely our biggest challenges in this country have been that we've just had so many people unvaccinated. If we had 90% of people vaccinated, we would really not be having any problem at all now. And it just hasn't worked out that way, which has been really sad. But like in San Diego, the numbers that we showed was, it was like last month there were 520 hospitalizations of unvaccinated people, and there were like 13 of vaccinated people. I mean, it's just, man, you want to talk about a single number that shows you how good vaccines are. Those are the types of numbers that
Probably the vaccinated people went home too.
Yeah. Oh, yeah. The vaccinated people, they all went home. Yeah, absolutely. All right. Well, yeah. Thank
you, Shane.
Yeah, thanks. Lots of fun.
Take care. Bye-bye.
Thank you.
Always great.
Yep. Clarity.
Yeah. And I'm just so impressed, like I said, with the immune system. Just thought of everything.
It's pretty fantastic.
It's amazing.
It's fantastic when it works. It doesn't always work.
That's true. I mean, sometimes, it does a little too much.
Or not
and then
enough.
I can't go outside
Some
when
people
there's
don't have
pollen.
it. Some people don't have it, yeah. Sometimes it causes disease that it shouldn't be. It's cool, but I think viruses are cool too. I got to fight back for viruses. Nobody wants to because they kill you, but they're really cool.
I kind of sort of wanted to ask him the ultimate hypothetical question. Maybe we'll get him another time, which is 10 years from now, are we going to be vaccinating people against SARS-CoV-2?
Yeah, I think that's very interesting and just to think about. What do you think, Rich? I know what I think. What do you think? Let's poll us. You think we're going to be vaccinating in 10 years? Just yes or no?
Yes or no?
Yes or no?
Yes.
Brienne?
Yes.
Kathy?
Yes. Vincent?
No.
So
Now
why do
can
you
I
say
qualify
no?
my answer?
I
don't think we're going to need to, but I think we will.
Okay, that's fine.
I think that we might need to, because I'm not sure what the percentages are going to be like for those who are immune.
I mean, maybe not in 10 years, but in 20 or 30 or 40, maybe. The only reason is because of what Ralph Barrick said back in February. He said, the common cold coronas probably did the same thing when they emerged from some animal reservoir. Well, we didn't know anything about pandemics thousands of years ago. And eventually they became benign-ish, and maybe that's because of large levels of population immunity, right?
Can you name anything that we've ever had a vaccine for where we now no longer vaccinate?
Smallpox, yeah.
Well,
and soon to be
so
polio soon to be, because we're eradicating it, yeah.
But you have to have a single reservoir for that.
But the history of immunization is too short to be able, because these viruses have probably evolved over hundreds of thousands of years to become what they are now. Maybe it's not even a viral change also.
Well, and I think the other question is, Shane mentioned this, that some of these situations where you're getting additional immune experience are giving you such breadth that you can neutralize SARS-CoV-1 in addition to SARS-CoV-2. What if we were to figure out a way to get breadth against more coronaviruses? That might keep us vaccinating and preventing other diseases as well.
I mean, I think it's a fair point that, we haven't seen that happen. I always use that in many levels to say, I've never seen a virus become more virulent, right? But I agree with that. But, I think we have a lot to learn from the common cold coronaviruses, especially, we learned in the past year that they do antigenic variation, which none of us thought they did. And how does that impact what happens in people? We really don't understand. So I agree it could go either way, but I'm not.
So many missed opportunities to have studied them in the past.
Really. It's true, because, and if you talk to the corona community, and the emergence of SARS-1, they all became sought after, and they said, up until now, nobody cared. they infect animals and they cause colds in people. Anyway, that's very interesting. Good stuff. And I think immunology, Brianne, to address your question, is learning much more than virology is right now, because you got to work on this bloody virus and a BSL-3.
Yeah.
And that's hard for the immunologist. Give me some serum and I'll do a lot. Or a fine needle aspirate.
Shane's approach to that was really interesting, and that we've never had an opportunity to look at this large a population in this kind of detail before.
I think flu has had, right? There, a lot of people get flu every year. And so there's a lot of flu immunology, but there's nothing like I don't know what, during a flu pandemic, the last one was 2009. I don't remember so many immunological studies. Do any of you?
No, I don't really either. And I don't remember as many kind of immunological insights or situations where you would now want to make sure that you show these data in a class or in a textbook, because it's such a kind of perfect example of whatever phenomenon. Whereas here, I want to put so much of the data we've been seeing right into my class.
Plus, that pandemic was one wave, right? Essentially.
Yeah, it was one wave.
Really quick.
And remember from Friday's excess mortality, very low mortality. I think 400,000 people died. Which is not zero. It's still, but it's it's not a lot. When he was talking about the size of spike, I was trying to think, how big is HA, and is it much smaller? And is that why there's a crappy T cell response to maybe flu vaccines or something like that? Does anyone know what the size of the HA is roughly?
I don't. I only know the size of spike.
1,273 amino acids. What is it? Length of influenza HA in amino acids. Let's see. What did I do without Google?
I don't know.
Up in textbooks.
We went to the library. I actually loved going to the lowest levels of the library. Nobody was there. It was all dusty, and you open an old volume from the 50s and you see what people were doing. I just loved that. That was cool, wasn't it? Well, I'm not finding it quickly.
Well, while you're looking, I was going to make another comment about influenza pandemics versus this one.
Yeah.
This is a completely new virus.
Yeah.
And, as time went on, the different manifestations of the viral infection, as the immune system, as he was talking about, as you go 3 weeks out and the disease gets more serious and now it's in your lungs, and then, all the things with the various types of long COVID and so forth. I mean, I think that lends itself to a lot of people wanting to know what's going on.
Sure.
And to my knowledge, influenza doesn't have that breadth of organ systems and the blood clotting and all those different kinds of things that we have with SARS-CoV-2 infections.
I think the avians do, but there's so few cases, right? It's very different. I'm not finding the bloody length, and people are sitting there screaming at me. I
have 566 residues.
Okay. Maybe roughly half the size of spike, huh?
Oh, yeah.
I mean, although I think that's the monomer, not the trimer, but I think 566 is what I'm getting.
Okay. I don't know if it explains anything, but anyway, let's do a couple of emails here. Brianne, can you take that first one?
Sure. Brea or Bree writes, hello, TWiV, and thank you for all you do. Flagstaff, Arizona, partly cloudy and 60 Fahrenheit. Questions for the COVID clinical updates. If you are fully vaccinated and exposed to COVID, does it give your immune system a boost because it's reacting to the virus? Same question, but if you were to have a breakthrough infection. I am, of course, not saying that one should intentionally expose themselves, but I'm wondering if the vaccine provides protection from severe illness, but community spread is still increasing. Would a happenstance exposure potentially help boost immunity for a little bit? Sorry if the question is not clear. Thank you for your time. Best of wishes, Brea, Bree, something like that. And I would say that we just talked a whole bunch about that. In theory, if you are infected, again, you would get a bit of a boost. Yes.
Yeah, we did discuss that with Shane. So I think a La Brea tar pits, right?
That's what I was thinking too.
But who knows? Who knows how people say it? So I was looking on the CDC website this morning, and I'm not sure if that's a good or a bad thing, frankly, but sometimes it's good, sometimes, but I do look at it, and they do not say there's evidence for transmission from vaccinated people who get infected. Okay.
Do they say there is not evidence for it, or do they not discuss it at all?
No, they say there's no evidence for transmission from infected vaccinated people. I mean, I think that's been implied in a lot of the discourse, but I'm not sure there's good evidence for it. I wondered if any of you had seen anything.
I mean, there's too many penguins on the iceberg.
Too many penguins. Okay. But this listener is just saying about transmission after vaccination. That's all.
Yeah. And I cannot cite a publication that demonstrates a vaccinated person being infected and transmitting the infection.
Okay.
But that doesn't mean it doesn't exist. Just means that I've got too many penguins on the iceberg.
No, she's not asking about transmission. She's just asking,
sure,
is it going to help you as an individual?
She says, does the vaccine provide protection from severe illness, but community spread is still increasing? That my understanding is that she's saying the spread is still increasing even if you're vaccinated.
No,
right?
I read that as, is she thinking that if you're vaccinated, but there's a lot of virus in your community, are you going to be, is that going to be a good thing for you to potentially get exposed?
Got it. All right. So I should have given you the next one, Brianne. I'm sorry, because
that's okay.
This is for you. Rich, you want to take the next one, please?
Sure. Judy writes, hi, Twivers. This one's for Brianne. So you listening, Brianne?
I am. I'm listening.
She and I have shared an interest in the Germinal Center story of SARS-CoV-2 infection. So I thought she'd appreciate this paper. As you know, there was a paper that came out back in August 2020 by Kaneko et al. That showed that people infected with SARS-CoV-2 did not develop germinal centers. Oh, that was a big, that was an immune episode, right?
I believe so.
Was it on immune? Okay.
I think it was immune.
I think it was covered on TWiV at the time, whatever.
Immune, yeah.
And she gives a link to the paper. Well, it is now almost exactly 1 year later, and another paper has just come out showing that persistent germinal centers do form in people vaccinated with the Pfizer vaccine. She gives a link to that, to Nature paper. Just more evidence that immunity from vaccination is not the same, and indeed is probably even better than immunity from natural infection. I love the show. So appreciative of all the things you do for our community. Stay well. Keep on Racaniello. Judy. Okay, so I'll leave it to, this is for Brianne. You got a comment?
So I think that it's a really interesting observation, but if you look at that original paper with the germinal centers, those were in patients who died. They did, they got germinal centers from patients at autopsy. And Shane mentioned some studies in non-human primates, and he also mentioned somebody else doing them in humans where they were taking the lymph nodes in live individuals. And it seems like they were seeing some good germinal centers in those cases. And that's kind of why I asked Shane about this, because you could imagine that maybe that was only something, that destruction of the germinal center was only something you see in a really severe case. So it's sort of hard to have that comparison. I've been really intrigued by what was going on there and how that can change the quality of the response, but I don't think that always happens during infection. And I think we need to look more at some of the studies that Shane mentioned about looking at germinal centers in living animals or patients. So it's not just the severe individuals.
I'm sitting here thinking that throughout this episode, we've been talking about germinal centers, and there may be a fraction of our listeners out there who are new enough so that that goes right over their heads. Could you, in 25 words or less, Brianne, tell us what
25
a
words
center
or
is?
less.
Yeah.
I'm going to start
I'll
counting.
give you 50.
Germinal centers are areas in a lymph node or in the spleen where a B cell goes after it sees its antigen in order to further develop. It can get better antibody production abilities, so it can change its specificity. It can sort of change in terms of breadth, and it can become a memory cell, and it can also learn to do some new and better functions. And so germinal centers are kind of the place where B cells mature further after they've seen antigen when they're in the lymph node.
See,
Higher education for B cells.
I was thinking of
In lymph nodes.
where B cells go to party, but you kind of threw water on that. Where B cells go to get mature, it's not partying.
I usually say that secondary lymphoid organs are where the immunology party happens. So
Good. Now, Judy said, more evidence that immunity from vaccination is not the same. And indeed, I would agree it's not the same as infection, but probably better than immunity from natural infection. I'm not sure that Shane would say that, right?
yeah, that's specifically why I was asking him about the quality.
Yeah.
And I think that, he did not support the idea that it's necessarily better.
Okay.
For those who want to know more, TWiV 161, Concerto in B, where we had Gabrielle Victoria on, and it was all about germinal centers. And that's where I learned what a germinal center was. It's just extraordinary.
It's natural selection for B cells.
Kathy, I don't know if you were on TWiV. When Brianne says quality, I just think of this old meme we had on TWiV years ago, where someone wrote in the koala tea from something, and it turns out it's a play on Merchant of Venice. The quality of mercy is not strained.
It's not strained, yeah.
The koala tea, and the guy wrote it to us as a joke, the koala tea, and it's koala as in the koala animals. Koala tea. Oh, what was it? I can't even remember the original TWiV. Let's see, koala tea TWiV. How about that? The koala tea of Marseille. No. Anyway, you were not on TWiV, but I just wanted to tell you because it's from Shakespeare, and the guy wrote it to us in a cryptic fashion, and we're like, what is this? What is this koala tea? And someone wrote in and said, you idiots, it's from Shakespeare.
Here's TWiV 81, the one we picked on last time.
Yeah,
Be a virus, see the world. The little the descriptor for that has the koala tea of Marseille. So maybe embedded in there somewhere. That was you and me.
It was just a funny episode before we really got popular, and there were just a few people listening. And because nowadays we have, maybe now people are going to weigh in on this because they know about it. I thought you would appreciate that, Kathy, being from a line in Shakespeare.
Yeah. No, and I remember having heard that.
Yeah,
it was before I was on the show.
Yeah, 81 was before BK, right?
Yeah,
before
and
Kathy.
it was even before the first one that I went to or heard, which was the one at Montana State. That was 92.
The first one you were on was when we visited Michigan, right?
Yep. Yeah, that was in 2011.
I remember you were driving me somewhere, and I said, "What are you going to do? Are you going to shut your lab down?" So this was 10 years ago, probably, right? And you said, "I don't know. I'll just keep listening to TWiV." And I said, "Well, why don't you just join it?"
And I don't remember that.
And you said, "I'll think about it." That's what most people say. Yeah. All right. Kathy, can you take the next one?
Dan writes, greetings TWiV team, and hello from Des Moines. High today of 33°C, tropically humid and still a bit smoky here. Not great weather for hanging from a rope cleaning windows, but someone has to do it, I suppose.
Wow.
We're about to have a giant state fair here, and after the not so great study on COVID in deer, I'm wondering about the potential for zoonotic spread. Just to set the scene, the Iowa State Fair normally gets over 1 million visitors. Very few people here are wearing masks. Daily cases are trending up, going from 50 or so per day a month ago to over 1,500 yesterday. So, oh, wow, that's a huge increase. Population here is around 3.3 million, with about 50% fully vaccinated. At the state fair, there are large display barns filled with sheep, goats, cows, pigs, and chickens, as well as other random animals. For example, they have a really big elk. The barns generally have pretty good air circulation, large doors that are kept open, big fans, etc., but they are covered and will be filled with people. What's the word on transmission to and from farm animals? To me, a guy that cleans windows, it seems like this might present a dangerous opportunity for a zoonotic crossover event. What does TWiV think? Regards, Dan. So, I think if we were going to have had spillover to farm animals by now, we would have heard a lot more about it. We have heard about some spillover to mink and cats and great apes in zoos and things like that, but we haven't heard too much about farm animals. So I'm guessing that it's maybe not going to be an issue. What do you guys think?
I think that with respect to SARS-CoV-2, the biggest danger here is the people. First of all, all these people with only half vaccinated in this small space sounds like a super spreader event to me. And I would say that with respect to zoonotic infection from either a SARS virus or any other virus for that matter, we've been doing state fairs for as long as humans have been around, and the risk is low to negligible. What I think is even more interesting is spillback, whether some of these creatures are going to pick up SARS-CoV-2 from the humans, okay, and whether that could seed some sort of event. I doubt it's seeding an event, but I wouldn't be at all surprised if a couple of creatures. Now, what did we have here for creatures? Sheep, goats, cows, pigs, and chickens. No tigers.
No tigers, no. No
Tigers and gorillas, They're
no
the ones
cheetahs or lions.
Yeah.
There have been no reported cases of pigs, horses, sheep, chickens, or cows getting COVID.
Yeah.
But they haven't been. This is from last August, and it says it's actually from the College of Food, Agriculture, Environmental Sciences at Ohio State, and they said, but nobody has checked them in the lab to see if they're susceptible. Must have been done by then. Since then, I haven't heard it for sure. So
I also think, Dan, thank you so much for writing.
it's a good point.
And thank you so much for listening.
Thank you for washing the windows.
Yeah, great.
Can you do mine? Yes.
For any kind of person, these are great questions and great logic. So don't sell yourself short here, Dan.
I think it's if I were going to this thing, well, first of all, I always see pictures of little kids kissing pigs at these fairs, and I think that's not a good idea. If I were going with a lot of people, I'd be wearing a mask.
Yeah. Oh, I'm not going to this event.
Right. I'm not going to go at all.
Mask or
But
not.
if I were somehow forced to go, and I can't imagine how that would happen, if someone offered me $1 million to go, I would go and I would wear a mask. And I would put the $1 million into microbe.tv, just so you know, folks, okay? I'm not going to keep it for myself.
I was just thinking about the reason why they have the doors open and the fans are probably because of the smells.
Yep.
Yeah.
Yep. Yeah, you bet. But I think in the next year, we're going to learn about a lot more animals,
Yeah.
including wild ones. I'm looking forward to hearing about that. All right, let's do one more here from Charles. Hello, TWivers. Another summer's day in North Carolina, 93F, 34C. A bit humid, some clouds. It may rain later today. This may bug me more than it should. But I'm sick and tired of articles and papers condemning antigen tests for SARS-CoV-2 because the authors are following the FDA's lead and using the wrong, quote, gold standard, end quote. PCR should not be the gold standard. Viral culture should be. See link 1 from the NIH, and he provides an article. Antigen-based testing, but not real-time PCR reaction, correlates with SARS-CoV-2 viral culture. Yes, from January of 2021. Interesting. The antigen test has demonstrated a higher positive predictive value, 90% than PCR when compared to culture positive results. I'm not surprised, actually.
I'm not surprised either.
I'm not either, but I'm cool to read that paper.
Yeah, we should.
Let's put that in the queue.
The latest example is from Germany, entitled, quote, Diagnostic Efficacy of Rapid Antigen Testing for SARS-CoV-2, the COVID-19 Antigen COVAG study. That's link 2. It's medRxiv. I found it at MedPage today with the headline, Roche Abbott COVID antigen test just so-so in real-world data. Unsatisfactory for screening asymptomatic people, say German lab professionals. Link number 3, that's from MedPage. So the medRxiv conclusion is the sensitivities are unsatisfactory. This calls into question whether their widespread use is effective. You know, that's funny because Thursday, Daniel reported on an evaluation of at-home antigen tests, and that was fine.
Well, does the medRxiv say that it's unsatisfactory, or does MedPage
the medRxiv says it's unsatisfactory? That's the conclusion.
Okay.
So then continue with Charles. What the hell do you want a screening test of an asymptomatic people to do? I would think you would want it to show if that person is infectious or not. Which test is better for that? Well, in most circumstances, it's the antigen test. If you're tested very soon after exposure, the PCR test would be better, but only for a day at most. If you're asymptomatic and non-infectious, would you want a test to show you are infected? Not me. Sorry for the rant. I have been an advocate for antigen testing for a long time, going back to before TWiV 640 with Michael Minna in July 2020, with a letter to TWiV in early June of 2020. It upsets me that an uneducated IT guy gets it and those doing the research don't. Okay, it really pisses me off. I can't imagine how you guys feel when you see so much bad research. Thank you for letting me rant, Charles. I think you got infected by the TWiV ranting, right, Charles? Yeah, this happens a lot, Charles, especially in a pandemic.
I keep thinking about my daughter's experience in London, where you just. As a matter of fact, I was going to use. Maybe I'll use this as a pick down the road. You can go to the National Health Service website and say, "Send me rapid antigen tests." Boom, they come to you in the mail. Yeah,
that's awesome.
You can have one. You can do. The closest equivalent that I know of to lick a stick. You can test yourself every day for free.
So I remember when the CDC Provincetown study came out a few weeks ago. Remember, vaccinated versus unvaccinated persons in this outbreak. The PCR viral RNA copy number was equivalent in both, right? And they got all worried based on that. But then, as Brienne pointed out on that episode, the Singapore study shows that viral RNA levels decline very rapidly in vaccinated people compared to unvaccinated people. At the same time, there was a paper, a separate study, where they looked at vaccinated and unvaccinated people who had become infected, and that's determined by PCR positivity. Over 50% of the vaccinated PCR positive people had no antigen positivity by one of these antigen tests, which to me suggested that they have little bits of RNA in them that are not infectious and not even making proteins probably. So I think the antigen tests are, as that first paper shows, they're very good indicators of infectivity. So there you go, Charles. Good job. And you're
nice rant.
You're just, we're all just something. Charles is just, but you're not uneducated, Charles. That's not correct to say that. You're educated in your field and perhaps more outside of that. Who knows?
Again, great logic.
It's good logic. Yep. Okay, let's do some picks here. Brienne, what do you have today for us?
I suppose I must be on some sort of little
thing?
arc or something. Yeah. So this is called How the Cat Gets Its Stripes. It's genetics, not a folktale. And this actually goes into showing details this is a New York Times article that talks about a paper where they were able to look at a gene that is responsible for the tabby stripe pattern. And are actually able to even see that pattern in embryos before the cat embryo has hair follicles. And so this reports on the research on that. There are some quotes here from Hopi Hoekstra, who does similar things in mice, who I think has been on TWiV.
Yep.
And it's just really cool and gets into, the genetics of how exactly this happens.
Looks like my cat there.
Also, it looks like my cat and Rich's cat.
So is this a segmentation thing?
It doesn't seem to be segmentation in the way that at least I learned segmentation.
Okay.
But I guess we have to ask somebody about sort of more specifics about how they mean segmentation. I think it's really just how this pattern forms. But it has that similar kind of gradient aspect to it that the segmentation stuff does.
So I got familiar with all kinds of cat genetics when I had a postdoc in my lab who is an international cat show judge and is very much aware of all the genetics of cats. And then just, for instance, the famous story about Siamese cats having a temperature sensitive coat color phenotype, so that at the tips of their ears, tails, and paws, is where they express the pigment. And so that's called pointed. But it's a tyrosinase missense mutation, I'm pretty sure, that causes that. So this is a continuing story on that line. It's really cool.
So when Hopi was on TWiV, she talked about her paper where they had these 13 striped squirrels, right? And her postdoc said, I want to slice the skin into thin strips and do transcriptomics on each slice to find the genes that make it colored and not colored. And she said, no way. There's just no way that's going to work. It was a Nature paper, right? They found the gene that controlled the striping. But she let him do it. It's good, right? That's you may not think it will work, but go ahead. As someone would say, sink yourself. But he didn't. It worked. It's cool. Kathy, what do you have for us?
So I have two things. One is a cool video. I think it looks like I misspelled it here. It's Kurzgesagt on viruses. And it's a YouTube video about giant viruses that's really well put together. I'm afraid to click on it because I don't want it to start, but with sound.
Try it and let me see if it actually starts, because I don't hear it.
I went there, but
I watched it.
I didn't hear your sound.
Kurzgesagt is a web page that is in a nutshell is what Kurzgesagt kind of means in German, and I did misspell it, but I'll fix it. And so the title is "This Virus Shouldn't Exist, but It Does," and it's had over 12 million views, but it's really well animated. And I only watched it one time, but I didn't find any flaws with any of the science in it. I don't know if you did, Rich.
I was watching carefully for them to make a misstep with this and talk about a fourth domain of life or something like that. My only gripe about it was that for me, they made too big a deal out of virophage. Because to me, those are just satellite viruses. Yep. They aren't that big a deal.
Yeah.
Okay, but
absolutely.
That's okay. It's an entertaining and actually quite highly accurate and educational video. It was nice. Well done.
Yeah. And then the second thing I picked, I just had to pick it. It was yesterday's astronomy picture of the day, and it's just beautiful. So it's from September 6th, and if you mouse over it. It highlights all the various things. There's a satellite trail and a green air glow and where Saturn is and where the Milky Way is and where the pine tree is, which was the reason for taking the picture. The idea was to photograph a statuesque pine in front of a central band of the Milky Way. And the plan worked. It's a time-lapse thing. Yeah. And they captured a firefly. Cool. It just makes this picture over the top, so you have to check it out.
A lot of flashes, 1, 2, 3, 4, like 20 flashes of the firefly. Cool.
I thought this one was absolutely beautiful, and I have to thank you, Kathy, for getting me hooked on APOD, because within the past week or so, there have been a number that have been just so gorgeous.
Yeah. This one's
So
really
I'm pretty sure Kyrgyzstan was a TWiV pick. So I searched Kyrgyzstan TWiV, and I found a letter from Peter in Wiesbaden, who wrote, P.P.S. Vincent, your pronunciation of Kyrgyzstan was absolutely delicious. So I said it right many years ago. Probably not anymore.
well, there's a Z in it, so I think it's Kurzgesagt.
Well,
Kurzgesagt.
he was German. He said it was delicious, and I probably said it wrong. Sometimes when people say there's something in another language in a kind of neat way, it's cool, right?
Yeah.
And he thought it was
Kurz
delicious.
is like short. Gesagt is like said.
Mm-hmm.
So in a nutshell, said short.
Mm-hmm.
Rich, what do you have for us?
So my justification for this being a science pick is that it's research, but it's journalistic research. I just read finished reading a book called Triangle, The Fire That Changed America by David von Draley. I got onto that because he's a columnist. I mean, he's had a number of different gigs, but he writes columns, opinions, and et cetera for the Washington Post. And I really liked the column he wrote recently. So I looked him up and I discovered this book, which it turns out is quite appropriate for Labor Day. We had a Heather Cox Richardson article the other day that starts with a triangle fire. This was a fire that happened in 2011 in the Garment District 1911 in the Garment District near Washington Square in New York City. That was sort of the breaking point for union demands and otherwise for safe working environments and et cetera. That everything went wrong in this, and it kicked off labor reform, and it also actually, based on this book, kicked off political reform that went far beyond that. One of the major investigators in the committee that investigated this fire was, now I'm going to blow this, Frances, what's her last name? Oh, she became the first woman cabinet member as Secretary of Labor for Franklin Delano Roosevelt. It's a great book and a really enormously scholarly effort. I don't know how people do this, research this stuff. So that got me to pick I guess he referenced this in the appendix of his book, a website that's from Cornell University called the 1911 Triangle Factory Fire. That's a web archive of everything you ever wanted to know about the Triangle Fire. That includes pictures and everything else. So this is a major event, and it's really a good example of how to find the truth, which I really enjoyed.
You know, I hate to say, but
Francis Perkins, thank you.
If this fire happened today, certain people would play it down and say, man, one of the reasons that as many people, 147 people died in this fire. It was on the 8th, 9th, and 10th floors of this building, the Ash Building, I think it was called. And one of the contributing factors was that one of the doors that might have been used as an escape on one of the floors was locked. And there was a criminal trial following the fire that tried to nail the owners of the building, the people who ran the company, for having that door locked, and they were acquitted. Okay, because the instructions from the judge to the jury was, you need to be convinced that the owners absolutely knew that that door would be locked at that time on that day. Yeah.
Okay.
Yeah, but he did. He locked it. He said he's locked it because he didn't want shirts stolen right from the factory.
Yes.
Well, it was going to be locked all the time. They should have convicted him.
So those raunchy people were around then as well,
Yeah, but I'm thinking of, massacre thingies that happen and certain people say it never happened or it's not that bad or blah, blah, blah. All this just try and politicize everything.
It could be. It could very well be that one of the reasons that this, had such an effect was that it was just so blatantly obvious. And people, a lot of the deaths were people jumping out of the windows because they didn't want to burn to death.
I agree. But when kids are shot in a school and nothing happens with gun control...
Yeah.
Right?
You're right.
I don't know if it was a better time. It probably wasn't, right? But at least something good came out of it, as the fire that changed America indicates. I just read her column this morning. That's why I'm thinking about this a lot. Okay, that's cool. Thank you. So my pick has to do with these two FDA people who are resigning, and I'm just curious about what's going on here. So I have a New York Times article, but you can find lots of articles about this. The Times' two top FDA vaccine regulators are set to depart during a crucial period. So Dr. Marion Gruber is director of the FDA Vaccine Office, and her deputy, Dr. Philip Krause, will retire. Gruber in October, Krause in November. According to an email that Marks, I guess he's the head vaccine guy at the FDA, Peter Marks, sent the staff members on Tuesday. And the article says one reason is that Gruber and Krause were upset about the administration announcement that adults should get a booster. We have two listener picks. Debbie, TWiV has become a necessary part of my podcast diet. I love eavesdropping on this very cool journal club. You have exploded my understanding of viruses and biology in general. I look forward to every episode. I thank you for all your time, effort, wisdom, humility, and good humor. And this pick is a website called River Runner. The site allows you to follow the journal of a drop of water.
It's really cool.
Any
It's
location
cool.
in the contiguous U.S. to the ocean. Using prodigious data sets from the U.S. Geological Survey, you can adjust the speed and detail of your journey, and you can pause anywhere along the way. Unfortunately, water that flows north through Canada stops at the border. That's funny. I hope someone takes up the challenge to do this for the whole North American continent. Yeah. Rabbit hole warning. Enjoy. Debbie is from Winnipeg. Oh, that's so cool.
That's awesome.
It's all based on the flow of water, right? Oh, you click on anything and you can see how everything flows. Neat.
Yeah, it gives you the local stream and then the river and then which lake or ocean it dumps into.
Wow, then it zooms in.
Yeah.
And the zooming is cool, and then it follows it. Wow. Why do rivers meander? I was thinking about this the other day when I was flying to Austin. Why do they meander? There's a reason for that, right?
Geology.
Beats me.
I mean, I think it has something to do with how long the river's been there, eroding.
Because, if you look at it's like you can see on this map there, they loop and then they go back and then they loop, right? Someone's going to write us and tell us. Okay. We have another one from Lynn. Good morning. Very early, 12:30 a.m. Clouds in a dark sky here in western Massachusetts. Damp from a day of drizzle. 61F16C. Just listened to the morning medical report from Kansas with you as guest. You are nothing less than terrific, and the program is worthy of a daily watch. It was just a bit disheartening to see Kansas not doing well with vaccination, despite this amazing show with these articulate doctors answering excellent questions. This should give you many more TWiV listeners, and that is cheering. I thought those doctors were really good. I was impressed. They were both upbeat, knowledgeable, good senses of humor, right? They were really good. I have a listener. Oh, by the way, in that Star Trek mask thingy, Kathy, that you had sent out with the Star Trek guys with the masks, he had found that or sent it with, don't be a red shirt, wear a mask. Is it true that on Star Trek, if the guys with the red shirts would get killed on the ground?
Yeah. Predominantly.
Yeah.
I think, Kathy, you dug up some data on that.
Yeah. So I think Rich put the figure into the Slack channel, and I just found the data that, yes, red shirts, 60% of those who died were wearing red shirts because of the role that they had in Star Trek.
Yeah, and I think that it's just also just sort of a general saying at this point of, being a red shirt is sort of being the extra one who's around, who's disposable.
So on this Kansas show, at the end of the episode, this one doc, forgot his name, he said, "Please wear a mask. Don't be a red shirt." And he put this slide up, and I had no idea what he was talking about. And then I saw it on the Slack. It was actually a few days before on the Slack channel. Okay, I have a listener pick, an excellent article from The Atlantic's Catherine Wu, who rivals Ed Yong in her science writings. Her thesis, waning antibodies doesn't mean waning immunity. Memory B and T cells are going to kick in to fight off the virus and prevent severe disease and death. It seems likely that for the majority of the vaccinated population who are not immunocompromised or very elderly, boosters should not be needed. I'm sending the link in a separate email. My phone is behaving a bit wonky. Best regards, Lynn, just a retired molecular biologist. It sounds familiar, huh? Waning immunity does not mean waning memory cells. Well, I heard that before. I heard that before.
I use Paul Offit's fire extinguisher analogy all the time for the immune system. I think that's great.
So I guess, I didn't put the link in. It was in a separate, but I think this is the correct link. Yeah. Yep. All right. I think we've heard from Lynn before because I recognize just a retired molecular biologist. That'll do it for TWiV 802, microbe.tv/TWiV for the show notes. You can send questions and comments to twiv@microbe.tv. And if you like what we do, consider supporting us. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker, Drew University BioProf Barker on Twitter. Thank you, Brianne.
Thanks. It was great to be here.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Fair enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.